Welcome to audio file number 3 for my CIC study series. Today we're going to start with antigen and antibody detection. Antigens are proteins of a virus or bacteria, while antibodies are compounds that develop in response to an antigen. Antigens are found on the surface of infective agents, and antibodies are found in serum. A typical example of a test is the enzyme-linked immunosorbent assay, often called ELISA, which has an enzyme that is linked to a particular antigen or antibody so that it can detect a specific type of protein associated with the infective agent. The direct fluorescent antibodies, or DFA tests, use antibodies tagged with fluorescent dye to detect the antigens. When tagged antibodies are added to a specimen and the antibodies attached to particular antigens, allowing for indirect identification when fluorescence is found upon microscopic examination. Each type of antigen has a particular shape, and only the antibodies that would match that shape can attach to it, causing the fluorescent reaction and indicating that an infection is present. However, some antigens have similar structure, and antibodies may attach to the wrong antigen, giving a false positive. The next section is covering urine dipstick test for leukocyte esterase and or nitrate. The urine dipstick test for leukocyte esterase and or nitrate is used as a quick and inexpensive test to identify purulent material or bacteria in urine. However, this test is most predictive if there are high levels of bacteria. The test for nitrate is more accurate than that for leukocyte esterase, and dipsticks that combine the two are the most accurate. While false positives are unlikely, false negatives may occur when testing for nitrates if there is diuresis that has reduced level of nitrates in the urine, inadequate nitrates in the diet, or infections caused by Enterococci and Acinetobacter, because these pathogens do not produce nitrates. Therefore, a high nitrate level is usually indicative of a urinary infection, but the absence does not preclude a urinary tract infection. Because of false negatives, a negative dipstick check should be confirmed through urinalysis that would include microscopic examination in the presence of there being symptoms in the patient. The next section covers urine samples. A urine sample taken from a patient who has not been catheterized should be a midstream sample. That is, that the initial amount of voided urine should be allowed to go into the toilet or the bedpan, and then the midstream catch is performed into a collection cup. The purpose of this is so that the initial flow of urine would flush out normal flora from the urethra. This is especially concerning about the bacteria that's growing around the end of the urethra. For a patient with an indwelling urinary catheter, urine for the culture should be taken from the catheter, not from the drainage bag. So if you have a indwelling catheter, such as a Foley, you should have a port on the catheter that's closer to the urethra that the urine can be drawn off of. When a urinary tract infection is suspected, samples for the culture should be collected in a cup with boric acid or stored at 4 degrees Celsius, because either procedure would inhibit the growth of contaminant bacteria until the specimen would get to the laboratory, avoiding the possibility of giving a false reading. The next section covers urinalyses. Urinalysis evaluates the following. Now, pH for urine usually would range between 4.5 to 8, with the average being 5 to 6. There are some medications, such as mandelamine, some foods, such as cranberries and vitamin C, that might make the urine more acidic. However, if you find that the urine is more alkaline, above 7, that would usually occur with something like bacteriuria, or a urinary tract infection, or possibly with kidney or respiratory diseases. Sediment is examined microscopically. Things that you would find would be red cell casts, which would be from acute infections, broad casts, which would be from kidney disorders, and white cell casts from things like pyelonephritis. Leukocytes that are greater than 10 per average per ml are present with UTIs, and you should never find any crystals in the urine. Also, glucose, ketones, protein, blood, bilirubin, and nitrates should be negative when you're doing a urinalysis. Urobilinogen should be 0.1 to 1.0 units. Urine glucose may increase with infection with normal blood glucose. Frank blood may be caused by some parasites and diseases, but also by drugs, smoking, excessive exercise, and menstrual fluids. Increased red blood cells in the urine may result from infections of the lower urinary tract. Urinalysis evaluates the following. Color is usually pale, yellow, or amber, and darkens when the urine is super concentrated. But some foods like beets, medications, stress, exercise, excessive fluid intake may also affect the color. Abnormal color may be caused by blood turning it red, bilirubin turning it yellow-green, high fever might turn it dark amber or orange, or diseases causing various color changes. Appearance should be clear, but may be slightly cloudy. While cloudy urine may indicate an infection with pus or blood present, it can also be related to foods, vaginal contamination, and degree of hydration. Odor should be slight, but foods like asparagus or medications such as estrogen may affect the odor. Bacteria may give urine a foul smell depending upon the organism. Also, specific gravity is usually about 1.015 to 1.025, but may increase if protein levels increase, or if there's fever, vomiting, or dehydration. Radiopaque contrast material, as well as dextrose infusions, may affect specific gravity. The next section is bloodstream infections. Bloodstream infections, sometimes abbreviated as BSIs, have increased markedly over the last few decades, with approximately 350,000 patients infected each year. BSI are defined as pathogens isolated in the blood of someone hospitalized for greater than 48 hours. There are two basic types. Primary infections arise in the bloodstream and may be related to intravascular devices. Secondary infections spread systemically from an infection somewhere in the body, such as from a urinary or wound infection. There are a number of issues related to diagnosing BSI from blood cultures. Skin preparation must be adequate to prevent contamination of a sample. blood volume must be 10 to 20 mLs in order to detect low concentrations of an organism. Timing should be as soon as possible after symptoms appear. Venipuncture should be done peripherally rather than obtaining blood from intravascular catheters, which may be contaminated with growth. The next section is culture media. Now, culture media come in different types. Differential medium is a certain type where an investigator may distinguish types of bacteria because of traits displayed in their growth patterns in that particular medium. Selective differential medium, now that's used in the isolation of Staph aureus, the most common virulent bacterial agent attacking humans. The medium contains a very high concentration of a material that the pathogen sought will tolerate, but inhibits most other bacteria. A fermenting material is then chosen that will further allow differentiation of the microorganism by the way it reacts. Enrichment medium. This uses a medium with a selective growth component, something that selectively resembles a nutrient from their own environment that only the microorganism in question can process. A selective medium has components added to it which will inhibit or prevent the growth of certain types or species of bacteria in the lab. In addition, or as an alternative, it may promote the growth of a desired species. One can also adjust the physical conditions of a culture medium, such as the pH and the temperature, to render it selective for certain organisms that are able to grow under these certain conditions. New selective culture media with enhanced selectivity are under constant development around the world. One recent one, for example, Turgitol, which is a trademark name, -4 in XLT -4 agar, would enable easier and faster diagnosis of Salmonella, a task which is often complicated by the presence of background flora and other Enterobacteriaceae on an agar plate. Chemical and pH changes within the medium allow Salmonella species, such as black colonies, to be differentiated from other organisms such as E. coli, which would show as yellow colonies, and Shigella, which would show as red colonies. The next section is collecting and culturing air samples taken in pharmacy clean rooms. Now, rules for collecting and culturing air samples taken in pharmacy clean rooms are as follows. Check engineering controls. Gather baseline data with microbiological sampling prior to activating unit when commissioning a clean room. Conduct air sampling on a regular basis to check for cleanroom microflora that have penetrated a barrier. Collect samples on nutrient media and incubate them to promote microbial growth. Attempts should be made to control or eliminate these variables as much as possible. Media selection and storage, plate handling, incubation temperature, species viability, and species competition. The next section is covering the United States Pharmacopoeia, often abbreviated as USP, and their recommendations for culture, plate media, and incubation. The USP recommends conducting sampling using non-selective malt extract agar or trypticase soy agar, abbreviated as TSA. When using malt extract for fungi culture, you should incubate at 20 to 25 degrees Celsius for up to 7 days. For bacteria, yeast, and mold, incubate at 22.5 degrees Celsius or 32.5 degrees Celsius for 48 to 72 hours. TSA media should be incubated at 30 to 35 degrees Celsius for 48 hours. Most importantly, the plate media, incubation temperature, and sample time should be consistent to ensure accurate comparison of data. The decision to change culture media should be phased in gradually and include side-by-side plate sampling for data comparison. Media should also be selected to assess specific microbiological species. An example, gram-negative or positive bacteria. The next section covers disease-producing microorganisms may be isolated from mixed bacterial populations. Isolate microorganisms from mixed populations by inoculating and streaking specimens directly onto agar-coated plates, selectively inhibiting them with antimicrobial drugs or aniline dyes, table salt, or other inhibitory agents, capillary electrophoresis, abbreviated as CE, using narrow bore fused silica capillaries, using capillary zone electrophoresis, abbreviated as CZE, to resolve bacteria into discrete electrophoretic bands, and employing selective medium containing lactate and inorganic salts is used in counting desulfovibrio in soil or water. Now, isolating and identifying many of the disease-producing microorganisms spread through fecally contaminated water is not always feasible. So, indicator organisms, coliforms or streptococci that are generally present in water containing the pathogens, are generally present in water containing the pathogens, are used to detect the possible presence of enteric pathogens. The next section covers transport media. Common transport media include, for biopsy, tissue, or swab, use viral transport media. For blood, use a EDTA, often a lavender top, or heparinized tube, often a green top. For CSF, or cerebrospinal fluid, or respiratory secretions, use a sterile container. For nasopharyngeal wash or throat wash, use a dry, sterile specimen container. For sterile body fluids, use a sterile, leak-proof container. For stool, use a leak-proof container with no preservatives. For a vesicle, place a swab in viral transport media and transport immediately on ice. For urine, a sterile container without preservatives. For anaerobic cultures, an oxygen-free vial, transport media with strong reducing agents, oxygen-free tubes, or plastic oxygen impermeable envelopes with catalysts that change hydrogen and oxygen to water. The next section covers clinical evidence, laboratory evidence, and supportive data. Clinical evidence derived from direct observation of the patient by infection control staff, supported by information obtained through chart review or review of other ward or unit records, and the possible addition of diagnosis of infection by a surgeon or physician making direct observations during surgery. Laboratory evidence comes from cultures, antigen or antibody detection tests, microscopic visualization methods, and the next section of this is supportive data, which would be derived from X-rays, ultrasounds, CT scans, magnetic resonance imaging, MRIs, radio label scans, endoscopic procedures, biopsies, needle aspirations, or other diagnostic studies. In cases where resolution of the problem is not immediately at hand, a thorough review of the medical literature may be called for. The next section is selecting and collecting specimens during infection control and outbreak investigations. Some key points to do here is to follow protocols set down for collection of each type of disease. Collect in amounts that are sufficient to allow effective and complete analysis. Obtain written consent from patients if specimens will be stored. Complete collection before dosing the patients with antimicrobial agents. Collection in viral infections should occur near the start of the acute phase to improve virus recovery. Avoid contamination, particularly by organisms from skin and mucous membranes. Keep specimens at required temperatures. Label all specimens from who they were taken, the date, the time, the location where they were taken, and who took them. Take care with biohazardous specimens that require special containment, and do not delay sending the specimens to the laboratory. The next section covers the collection and transport of respiratory tract specimens. This is covering a kind of a broad or general term for what you're collecting, and then kind of what you would be using to send that in. So for like a lung abscess or emphysema fluid, you would want a container for anaerobic transport, vial, something like that. For transtracheal aspirate, you would want to use anaerobic transport vial. For TB or tuberculosis, you want to use a container. Well, it really depends on what type of tuberculosis sample you're getting. If you're getting a sputum culture, you want a clean, leak-proof specimen container. They're going to be single-use, 50 ml capacity usually, and you're going to have the patient kind of first thing in the morning provide a sputum sample where they're going to cough before they brush their teeth or anything else. They're going to cough, as hard as they can to provide a sputum sample, and then spit that sputum into the container so that can be sent in for testing. It says here that sputum analysis is the most dangerous lab procedure. It must be done in open air at a distance from other people. Never collect it in the lab itself. That really depends on the setting, time the patient's been on medication. That's individual to the scenario as well, just so you know. Next is bronchial washings. You'd want to use a container transport device that is sterile with a screw cap. For mycobacteria, you'd want to use a sterile container with a screw cap. the Mycobacterium, you know, you're usually going to be collecting that in the same fashion as the tuberculosis, early morning, deep cough, sputum. Then the nasopharynx samples are going to be a sterile container, usually having either a wire, flexible swab or something like that to take a sample in the nasopharynx, and then that is either broken or bent off into the container. For sinus aspirate or a tympanocentesis, you're going to want a sterile container, anaerobic transport vial. For the oral cavity, you'd use a container transport device with a swab inside of it. So the swab will be used to swab the inside of the oral cavity. For a throat culture, you'll be doing a swab, again, container. You'll have to make sure that when doing a throat culture that you're avoiding, you know, other infections that might be there, and getting the swab to, you know, what it is that you're trying to swab. And that's true for any of the oral or throat samples that you might take. And then we have, lastly, the tracheal aspirate. So you'll use a sterile screw cap container for that one as well. The next section is covering the collection and transport of clean voided midstream urine or urine obtained by catheterization or cystoscopy for viruses, C. Trachomatis, or bacterial. So for routine analysis, the microscopic evaluation of urine, sterile screw cap tube or jar is the container device, and the volume of collection for this would be 1 to 10 mLs. You would instruct the patient to discard the first portion of the urine stream and when voiding, and before voiding is complete, to collect the remaining portion of the urine in the midstream catch. And they would be doing the collection in the sterile container that was provided to them, and it would be best to have a clean catch. They will cap and label the specimen, or you will, depending on the situation. It's then placed in a refrigerator and held there until the courier picks it up. 24-hour specimens must not be collected, it says. Oh, that's probably referencing a 24-hour urine measurement, which is sometimes done in a hospital setting. The specimen is not appropriate for anaerobic cultures, viruses and bacteria. The C. trichomatis, the specimen is appropriate testing only for men. The next section is going to be covering the collection and transport of blood specimens. So for viruses, the volume of blood that you want to collect is between 5 to 10 mLs, but 30 to 50 mLs if you were testing for HIV. You'd want to send these in heparinized tubes. For bacteria, you'd want a volume of 20 to 30 mLs for an adult and 1 to 3 mLs for infants and children. You'd want to collect this blood in blood culture bottles containing broth or lysis, centrifuge lysing tubes. Additional considerations here would be to collect 2 separate blood samples during a 24-hour period with intervals between the cultures determined by the urgency of the clinical situation. For leptospires, you would want to collect 1 ml blood. The container you'd use for that is a heparinized tube. Other considerations for leptospires are to collect 2 separate blood samples during a 24-hour period, depending on the urgency of the situation for timing. For brucella and fungus, you'd want to collect between 20 and 30 mLs for an adult and 1 to 3 mLs for infants and children. The container you would want to use is once again, the centrifuge lysing tube or biphasic blood culture bottles. Additional considerations here is to collect, once again, 2 separate blood samples during 24-hour periods with a time depending on the urgency. For Borrelia, you would, says it doesn't really have a preferred volume. You'd be doing a peripheral smear, and that would be a wet mount. It should be examined by dark field microscopy or via a smear stained with aniline dyes. For malaria, again, you don't have a volume that is necessarily preferred. You'd be doing a thick and thin film on a clean glass slide. For filaria and trypanosomes, you'd want to get at least 5 mLs. You would use a sterile tube containing an anticoagulant, such as a citrate, oxalate, or heparinized. And considerations here would be a drop of blood should be collected on a wet mount, or it should be concentrated hemolyzed blood rather than stained thick and thin films is preferred. The next section covers the collection and transport of urinary tract specimens. Now, all except for suprapubic aspirates, which is collected and transported in an anaerobic vial, clean voided midstream urine or urine obtained by catheterization or cystoscopy for mycobacteria, fungi, suprapubic aspirate, and voided urine for parasites go into a sterile screw cap, tube, or jar. Though the volume of a sample for detecting suprapubic aspirate is not important, care in handling is, as analyzing this sample is the only certain method for establishing a diagnosis of anaerobic bacteriuria. More than 20 mLs should be sent to the laboratory immediately upon collection or refrigerated for the detection of mycobacteria and fungi, but not held for more than 24 hours, as specimens that are that old are not usable. For detection of Schistosoma hematobium eggs, embryonated eggs of human schistosomas or blood flukes or Miracidia, all very uncommon in North America, submit a 24-hour collection of voided urine. The next section covers the collection and transport of fluid specimens. So for exudates, transudates, drainage, and pus, you'd want to collect between 1 and 5 mLs. You'd want to use a anaerobic transport vial. For collection of abdomen or chest fluid, again 1 to 5 mLs, but for bacteria, and less than 10 mLs for mycobacteria or fungi. You would use anaerobic transport vials for those. For synovial fluid, again 1 to 5 mLs for bacteria and less than 10 mLs for mycobacteria or fungi. And you would use an anaerobic transport vial. If you suspected gonococcal arthritis, then you could use a Thayer-Martin medium modified as an inoculate for the synovial fluid. And for CSF or cerebrospinal fluid, you would collect 1 to 2 mLs for bacteria or viruses and less than 2 mLs for mycobacteria or fungi. And you would collect that in a sterile screw-capped tube and immediately have that sent to the laboratory. The next section covers collection and transport of gastrointestinal tract specimens. So for stool and bacteria, the volume isn't necessarily important. You would want a container that's a sterile screw cap tube or container, and you may want a transport medium swab depending on the type of bacterial culture that you're retrieving. For stool, for ova and parasites, once again, the volume isn't necessarily a big factor here, but a stool carton sealed in a plastic bag with a polyvinyl alcohol preservative is important. For anal swabs for pinworms, again, the volume not necessarily important. A sterile plastic swab in a tube is what is used. A collection is accomplished by the swab of the perianal area, preferably in the morning upon rising and prior to bathing or defecation. And for a stool culture for viruses, you would, again, volume not necessarily important, but you'd be using a sterile screw cap jar or tube, or a transport medium swab for collection. The next section is the collection and transport of catheter specimens. For Borrelia, you would use a sterile screw cap tube or culturette. Additional considerations here would be that when collection is performed, the skin entry site should be disinfected, the catheter removed, and the end going into the tube should be clipped off. Then we have for suction or drainage specimens, you would use the volume, not important necessarily, and you would use a sterile screw cap tube. For cultures, the volume, again, not very important for any of these in this section. You would use a swab or anaerobic transport vial. For scrapings or dermatophytes, you would use a sterile petri dish for the culture specimen. The next section is collection and transport of specimens of sexually transmitted diseases. So here for Treponema pallidum, you would use a container that would be a serous exudates on a clean glass slide or in a capillary pipette. Additional considerations: the lesion should be abraded with a clean, dry sponge, and the preparation examined by dark field microscopy immediately upon collection. For chlamydia, you would use a container with a swab held in a sucrose phosphate solution, sometimes abbreviated as 2SP. Additional considerations for chlamydia would be that the urethra or cervical material should be extracted on a swab in solution. Then, if stored, it should be refrigerated. For ureaplasma, urealyticum, you would use a swab held in a sucrose phosphate solution, 2SP, and considerations here. Again, the urethra or the cervical material should be extracted on a swab in solution, and then if stored, it should be refrigerated. For Trichomonas vaginalis, you would use a sterile swab, and no other special considerations are noted here. Two other things I want to point out about STD collections here. One is about gonorrhea, and for gonorrhea, the transport device is either a swab or a petri dish with a modified Thayer-Martin medium containing antibiotics and nutrients which facilitate the growth of gonorrhea species while inhibiting the growth of gram-positive organisms and most other bacilli. The volume of collection is not applicable. Now for women, the collection is accomplished by taking a cervical culture. If the cervix is not accessible, the sample is taken by inserting the swab via the urethra or the vaginal canal. For men, collect the material for a smear and culture with a swab or sterile bacteriological loop should be inserted through the urethra. And the other thing I wanted to point out as a special note for STD collection is for Haemophilus ducreyi. Now I'm probably butchering that second part there, but the only thing pointed out here is that a swab should be used, a sterile swab should be used, and no particular volume is of need, a specific applicable amount. The next section covers the collection and transport of specimens regarding eye cultures. So corneal lesion or scraping samples, you would need, a lot of these where you're taking a sample where you are either scraping or rubbing up against something, you're using a swab or something else. There's not necessarily going to be any volume because what you're trying to do is to pick up the material onto the surface of the swab or something else that's then going to be used to transport that to the laboratory setting for putting it into petri dishes. So again, volume doesn't really pertain here, but for corneal lesions or scrapings, the specimen should be inoculated directly onto proper media and applied directly to a clean microscope slide for staining and microscopic examination. For conjunctiva examination, eye secretions, prostatic secretions from the eye would be collected in a container. Considerations for this is that the specimen should be inoculated directly onto proper media and apply directly to a clean microscope slide for staining and microscopic examination. For gonorrhea, a modified Thayer-Martin medium, different brands might be used for that, would be used, and additional considerations is that the swab should be inoculated directly onto the medium. For chlamydia, a sucrose phosphate solution, 2SP, or a swab should be used for a direct smear. With chlamydia, the specimen material should be extracted onto the swab in a solution and then refrigerated if stored. For bacteria, fungus, and viruses, you'd want to get the best sample that you can. You'd be using a sterile screw cap bottle, and the specimen must be collected. The sample that is collected must be an adequate portion to ensure that you recover sufficient organisms within the volume that was obtained. The next section covers the collection and transport of specimens of the genitourinary tract diseases. The first one being herpes simplex virus. So you would use a swab, and you should avoid calcium alginate. The specimen should be refrigerated for storage before testing. For testing of cervical or vaginal discharge, you would use a swab, and the specimen is not appropriate for anaerobic cultures. For caulocentesis fluid, you would use an anaerobic transport vial. For abscess testing of the pelvic, tubular, ovarian regions, you would use an anaerobic transport vial. For prostatic secretions, you would use, I'm just now noticing that the term seems to have jumped around on the paperwork that I'm studying, relating to the prostate fluids. So I'm seeing that actually is copied down incorrectly into the conjunctiva section that I just covered for eye cultures. So please disregard that. The prostatic secretions should be collected and sent in sterile screw cap bottles. Now I'd like to talk about the next section, which is covering swabs. Swabs may be used to collect specimens from the nares, throat, or wounds. If purulent material is being collected from a wound, it is better to send a sample of the pus in a sterile container rather than collecting a specimen with a swab if the drainage is sufficient. Now, with swabs, the collection should be done wearing gloves and holding the container that receives the swab as close as possible to the swabbed area to avoid contaminating the environment with droplets. The container should be labeled before collection. The swab is inserted into the container, usually a tube, slowly to avoid contaminating the mouth of the container. Some types require that the end of the swab be broken or cut, which has the potential for spreading infection, so it should be done gently. And prior to collection, you should look at or observe the actual swab. Does it have a spot where it breaks intentionally, or does it not? Do you need to plan ahead before using it for a collection? Always very important, and it has been a real-world struggle for me when doing field collections for outbreaks. And next section for swabs, under swabs, is going to be for the handling So the handling of the specimen container should be after the container is bagged. That's why you would get all the information filled out on the specimen container before doing the actual collection. So that way you have as little touching of the container as is possible. Now the transport should be done immediately, and the storage should be in designated areas following the guidelines for the type of test that's being done. Some refrigerated, some not, that sort of thing. Now on to the next section, which is urine specimens. Urine specimens should be collected following standard procedures. Urine specimen containers should hold at least 50 mL and have a wide base. They should be sterile, leak-resistant, break-proof, or as close to that as possible, and have a secure lid. Containers are used only one time and then discarded with other biohazard material. Collection may be from straight catheterization, drainage from a Foley, or obtained from urination, as we've discussed previously, through a clean catch method. The staff handling the container and the patient should be instructed in proper methods for obtaining the specimen, and anyone who is dealing with the container should wear gloves. Containers should be labeled properly, not on the lid, which might become separated from the container at some point, either during collection or after and during transport, or even after transport at the lab. The transport should be done with the specimen container inside of a secondary container, such as a Ziploc bag, so that the urine that might come outside of the container doesn't spread contamination to other samples. Storage should be in a designated area and inside the transport container. The next section covers aspirants. So an aspirant includes any fluids aspirated from the chest, the joints, the CSF, the sinuses, from abscesses, cysts, or any other magical, disgusting thing that a human body has decided to produce extra fluid in. Or even if it doesn't produce extra fluid, I should say, because that's always a possibility. So collection requires aseptic technique, including gloves, with a needle puncture, and then aspiration of fluid. Skin antiseptics are used, but those with a strong residual effect, such as chlorhexidine, should be avoided. The container should be dry, break-proof, and sterile. A protective mask is usually not needed, although it may be used when obtaining CSF and with some infectious diseases, such as meningococcal meningitis. The needle should be removed if there is a safety method to prevent needle stick, in order to reduce spraying the fluid. Gently expel into the specimen container, which is then bagged. That felt a little bit rough, so let me just repeat part of that, because the needle should be removed in a safe way to prevent needle sticks. In a hospital setting, this is always a major concern, especially when a patient's in pain, yelling. They might move very quickly and someone jerks back and accidentally sticks someone. And then after removal safely, it should be placed into a specimen container without spraying. The next part of aspirants is handling, which should be done with gloves until the container is bagged completely. The transport should be done immediately to the lab, and storage should be in a designated area following the guidelines specific for the type of the aspirant that was collected. CSF fluid degrades quickly and should be tested immediately. The next section is covering sputum specimens. The sputum specimens can easily contaminate the outside rim of the container, as people often cough directly over the container or place the container up to their mouths to cough into it. So, containers should always be considered contaminated. For the collection should be done wearing gloves if obtaining the specimen directly from the patient and when handling the container. If the patient collects a specimen, the patient should be instructed on how to do so properly. The container should be wide-mouthed and hold at least 50 mLs. If possible, the rim should be wiped off before the lid is applied and the tissue placed in the biohazard waste receptacle that is used to wipe it off. Collection should be done outside at a distance from other people and not in the laboratory. Handling should be minimalized. The specimen container should be placed inside of a Ziploc bag that is labeled with the name, date, and time of collection. Transport to the lab should be done immediately. Storage should be in a designated area, following guidelines for the type of specimen that is being collected. The next section is blood specimens. Blood should always be considered hazardous and potentially infectious. Collection should be done using antiseptic techniques and sterile equipment, with the person collecting the sample wearing gloves. Collection should be done using vacuum tube blood collection devices. Avoid the use of needle syringes if possible, or if blood must be transferred from the syringe, care should be used to avoid spraying blood or leaking droplets. In some cases, protective eyewear may be used. Blood should not be collected from an arm into which IV fluids are being administered, as this can dilute the sample. Blood is drawn in a particular order if multiple tubes are used so that there is no cross-contamination with additives from the tubes. Handling should include mixing the additive with the blood by rotating the tubes back and forth. Transport to the laboratory should be done immediately, and storage should be in designated areas following guidelines for the type of testing that is to be done. The next section is covering fecal specimens. Fecal specimens should not be contaminated with urine, paper, or toilet water, especially if they are to be examined for a microorganism. Collection should be done after the patient has urinated. If possible, the specimen can be collected in a clean bedpan. Plastic wrap is sometimes placed over the back part of the toilet to catch the feces, but the plastic can become easily contaminated. The fecal specimen is placed in a sterile container, preferably one with a scoop in the lid to facilitate the easier transfer. About 10 mL should be collected for viral or bacterial testing. Testing for parasites requires a larger specimen. The person handling the container and specimen should wear gloves. Swabs of fecal material are almost never sufficient for adequate testing. When handling should be done after the specimen container is bagged with label indicating the name, date, and time of collection. The transport should be done as soon as possible to the lab, and storage should be in designated areas following the guidelines for those tests. The next section covers pseudo-infections and pseudo-outbreaks. Pseudo-infections should be suspected when an organism is cultured from an unusual body site, or an unusual organism is cultured from any site on the body. In both cases, there is colonization of pathogens without the clinical signs of infection appropriate to that organism. The colonization may be superficial, the specimen may have become contaminated, or there may have been a laboratory error. When clusters of the same type of colonization occur without an infection, it is deemed a pseudo-outbreak. There are different types of pseudo-infections. Pseudobacteria, especially Bacillus, Pseudomonas, and Streptococcus species, usually as a result of normal flora or contamination of the specimen, is one type. Pseudomeningitis, with bacteria or fungus in the CSF, but without expected clinical symptoms, matching the organism, usually results from contamination of the specimen. Pseudopneumonias, usually related to contaminated bronchoscopes, respiratory equipment, or solutions. And the last type here is pseudobacteriuria, usually resulting from contamination of external urinary drainage systems. True infections must be differentiated from pseudo-infections so that patients can be properly treated. The next section covers molecular strain identification techniques used for outbreak investigation. Biotyping involves identifying different biochemical reactions. There are multiple methods, such as identifying the ability of a microorganism to ferment sugars. There are kits available for typing of different microbes. However, biotyping does a poor job of distinguishing among strains. Bacteriophage typing uses bacteriophages, which are viral intracellular parasites with the ability to infect bacteria by entering, multiplying, and destroying or lysing the cell. A series of bacteriophages with different ability to lyse a cell is used to differentiate strains of bacteria depending upon the microorganism's susceptibility to the lysis. This technique is time-consuming and not widely available because stocks of phages must be maintained. This test is most useful for identifying staph strains. That's Staphylococcus strains. It has been replaced more recently by DNA-based genotyping methods. Bacteriocin typing involves typing or identifying different antibacterial toxins that are produced by an individual strain of bacteria. This method is rarely used currently. Protein typing determines differences in proteins made by strains of bacteria. Pulse field gel electrophoresis, often abbreviated as PFGE, is a method of typing that separates large DNA strands. A typical technique is to apply 3 pairs of electrodes to a gel plate containing DNA produced by restriction enzymes that break the DNA into small numbers of large DNA fragments. The plate is covered with dye. A current that changes directions in a regular pattern is applied to the gel until the dye has spread across the gel. Then a solution is added that binds to DNA and causes it to fluoresce. Changing the direction of the current allows smaller DNA fragments to move more quickly than larger fragments, so there is a separation of long and short strands, providing the typical DNA band profile. Different organisms have different band patterns. Organisms are considered distinguishable if they have more than 3 bands in different positions. This typing method is very accurate, especially for Staphylococci, for Enterococci, and for P. aeruginosa, which I probably butchered that name as well. Restriction endonuclease analysis uses restriction enzymes to identify bacterial strains. Restriction enzymes are particular bacterial proteins that would recognize unique chromosomal DNA and plasmids, which are circular double strands of DNA that can replicate in a cell but are separate from chromosomal DNA. Restriction enzymes cleave or split the DNA at specific sequences. The fragments that are cleaved can be separated by size to produce a restriction endonuclease profile. This method can detect small differences in bacterial strains. There are 3 different types of restriction enzymes that can be used. Type 1 can recognize unique chromosomal DNA but cleaves the strands at random, at least 1,000 base pairs or BPs, from the site that they recognize. Type 2, the most commonly used restriction enzyme in recombinant DNA methods, recognizes the unique site and cleaves at that point, producing predictable strands. And type 3 also recognizes the unique chromosomal DNA but cleave at least 25 base pairs away. This method is useful for bacteria, viruses, and parasites that don't have plasmids. DNA hybridization or genetic probing involves a process in which DNA is denatured, which means heated to a temperature that allows the DNA to form into single strands. As the DNA is cooled, it recoils and can be joined to a complementary probe of labeled DNA, usually with radioactive phosphorus, forming a hybrid, which is then measured. Essentially, a DNA, or sometimes RNA, strand is marked chemically or radioactively with a substance that will bind to a particular gene, allowing this gene to be identified and isolated. This typing technique can profile similarities in sequencing among various DNAs, as well as the repetition of sequence in one DNA. Genetic probing is used to find specific fragments of DNA, referred to as target DNA. This form of typing does not require a viable pathogen and can identify individual strains of bacteria. It can also identify pathogens such as rotavirus and chlamydia, which are difficult to cultivate. However, it is a slow process and often not as sensitive as doing a culture. Plasmid profile analysis involves the development of a plasmid profile. The plasmid DNA is isolated from a microorganism. Then the plasmid DNA is separated by agarose gel electrophoresis. Plasmids can separate from one species or strain to another through a process called coagulation. So in some instances, an outbreak can be traced to plasmids rather than to a bacterial strain. However, this mobility of plasmids interferes with analysis. Analysis is improved by using restriction endonuclease enzymes to digest the plasmids and create fragments that are then analyzed by electrophoresis. Plasmid profile analysis can be applied to many different bacterial strains and can be completed within one day, but epidemic epidemic strains may not contain plasmid or strains that aren't. Sorry, let me back up. But epidemic strains may not contain plasmid or strains that aren't part of the epidemic, may contain the same plasmid profile as the epidemic strains. So, while plasmid profile analysis is a rapid and inexpensive technique for typing, it is not always accurate or sufficient to get the desired results. Polymer chain reaction, also abbreviated as PCR, makes millions of replications of specific DNA sequences without a living microorganism, I should say. When cells divide naturally, polymerase enzymes copy the DNA found in each chromosome by separating the two strands of the DNA helix so that each strand may be copied and then replicated. This process also requires the 4 nucleotide bases. Additionally, a short sequence of nucleotide primase called a primer is needed to begin the replication process. This can be done within a test tube, which is heated to separate the DNA strands and then cooled so that the primer can begin the process, and heated again so that the polymerase can begin to make copies using the nucleotides. This 2-hour method can be used to replicate and detect more than 22 different microorganisms that fail to grow well or at all in cultures. It can detect coding genes for toxins, virulence, and antimicrobial resistance, making this an effective tool for infection control. Ribotyping is a type of DNA fingerprinting analysis that utilizes genes that provide code for ribosomal ribonucleic acids, or rRNA. Various proteins along the ribosomal RNA comprise the ribosome, which is a structure within the cell in which the proteins are manufactured. The ribosome uses coding from the rRNA to place the correct amino acids in proteins. The genetic coding for rRNA varies more between bacterial strains than within them, so ribotyping distinguishes between bacterial strains. Restriction enzymes are used to cleave the genes that code for the rRNA into fragments, and then the electrophoresis is used to separate the fragments. Genetic probes are used to highlight fragments of different sizes so that they appear as bands in the profile. Different ribotypes have banding patterns that correspond to each type. Ribotyping has been successful in typing a variety of microorganisms, including strains of Salmonella typhi, E. coli, Campylobacter, as well as others. The next section is limitations and advantages of types of tests used to diagnose infectious processes. Though imaging may be used to research the effects of a given pathogen or toxin on the human body, for clinical studies or in response to an outbreak, laboratory tests of blood, stool, or sputum samples are the investigations of choice. Most responses are rapid, though in some cases incubation becomes necessary, slowing down the process of establishing the source of the illness. However, testing often fails to contribute to the diagnostic process while increasing patient risk and the expense of medical care. Modern laboratory tests and imaging studies are often mistakenly stressed, while history and physical examinations are downgraded as effective tools of clinical assessment. The next section covers culture contamination. Anywhere from 10 to 60% of all cell cultures may be contaminated with mycoplasma, which, because of its minuscule size, can pass through filters used to sterilize tissue culture reagents. Mycoplasma contamination may never be eradicated since they are immune to the usual antibiotics that work by attacking cell walls, something which mycoplasma lacks. Tissue cultures are vulnerable to contamination through a number of routes. Some of them would be careless handling of pipettes or flasks, dust from skin, hair, or clothing, aerosols from talking or coughing, dirty or ineffective sterilized storage containers, or other instruments. Now, often the contamination is not immediately apparent, lacking visible turbidity or changes in pH, and erroneous data can be the result because prokaryotes change nucleic acid synthesis, membrane antigenicity, growth rate, interferon production, enzyme kinetics, and cellular metabolism. The next section covers rapid confirmation of a communicable disease. Positive confirmation of a communicable disease like whooping cough, Legionnaires, bird flu, tuberculosis, or SARS is gained by culturing samples taken from patients who are suspected of being infected. Waiting to see if the bacteria grow can take weeks. There are now newer, faster ways of establishing confirmation through sensitive molecular tests. However, they are in their early stages and are proving that too often they are producing errors. Many of the new molecular tests give rapid results but are technically demanding, and each laboratory may do them in its own way. There are no good estimates of their error rates, so that should play a warning sign to anyone who's opting for brand new types of testing. But their rates vary in sensitivity and make false positives likely, so that also is a warning sign when using new tests. When hundreds or thousands of people are tested, as occurred in a recent suspected outbreak of whooping cough in New Hampshire, false positives can make it seem like there is an epidemic. When the biological culture reports came back months after the apparent outbreak, results were negative. All the apparent cases had likely been cases of common upper respiratory infections. There is no report of what expenses were run up by this error. This would be a good opportunity for me to interject a personal story of, or I should say experience, that working in public health, we often respond with a lot of manpower from local public health, state public health, hospitals, physicians' offices, and in some cases it's an extreme amount of time and money being spent to sometimes days or weeks later find out that it was not a case. It was not a case of someone who was indeed infectious. So it's always a possibility, but it's better to be safe than sorry in most cases if the evidence is supporting. The next section covers normal flora. The normal flora of the body is necessary and benefits the host in a number of ways. We're going to talk about some of those ways now. Now, in synthesis and secretion of vitamins, bacteria are an important source of vitamin K and vitamin B12, which is secreted by enteric bacteria such as E. coli. In terms of resistance to colonization, the normal flora attaches to host cells, preventing competing bacteria from attaching and colonizing. They also compete for nutrition. In terms of inhibition or destruction of pathogens, bacterial flora may produce substances such as fatty acids or bacteriocins, which could destroy an invading pathogen. In terms of developing tissue, in response to normal flora, the walls of some structures, such as the cecum and some lymphatic tissue, thicken and strengthen. As far as stimulation of antibodies, the normal flora have antigenic properties that stimulate formation of natural antibodies in the host. These antibodies can cross-react with the antigens of related pathogens as part of the immune system. The next section covers clinical classification of bacteria. In the clinical classification of bacteria, the classification takes into account those characteristics that are helpful in identifying infectious processes. We're going to talk about some of those now. So in gram-positive or gram-negative statuses, most bacteria are gram-negative stains, which would be red, or they would be gram-positive, which would stain as purple, although a few cannot be identified by staining. While gram stain isn't used to identify bacteria, it's frequently referred to clinically. When it comes to taxonomic status, taxonomy is based on the genera and species of a bacterium, but this can be confusing because some names have changed or two names might be used. The genome sequencing should standardize identification. When it comes to anaerobic or aerobic status, some bacteria are strictly anaerobic, but very few are strictly aerobic. Those that have flexibility and can grow in either aerobic or anaerobic conditions are called facultative. When it comes to the usual environment, bacteria are classified according to where they usually reside as flora or where they usually cause an infection. When it comes to virulence factor, bacteria are very widely in virulence. Some are actively invasive, but others only cause opportunistic infections. The next section covers gram-negative bacteria. The cell walls of gram-negative bacteria are characterized by red staining. The cell wall is thinner than that of gram-positive bacteria. However, there are two separate layers to the wall. A thin inner layer of a peptidoglycan and intervening periplasmic space, and the outer membranous layer, which is the lipopolysaccharide layer, which produces endotoxins, making gram-negative bacteria extremely pathogenic. A component of the outer layer is called the S layer. It aids in adherence and protection from pathogens. The outer layer serves to protect the gram-negative organism from antibiotics or detergents that would disrupt the inner peptidoglycan layer and provides resistance to penicillin and other compounds. Ampicillin is able to penetrate the exterior wall through many bacteria that have become resistant. The next section covers common gram-negative cocci, or round, bacteria. This is just a list of 3 common types. We have Neisseria gonorrhea, we have Neisseria meningitis, and we have Moraxella catarrhalis. Probably butchered that, but I don't say that very often. Next section we have is common gram-negative bacilli, which would be rods. And some common types here would be Haemophilus influenza, Legionella pneumophila, Pseudomonas aeruginosa, E. coli, and Heliobacter pylori.